Yeah, I'm gonna film what we do, so I'll post the video of the review session on the Moodle site so you can have access to that.
Are the answers for that problem set on Moodle 2? Are they going to be posted?
I'll generate them later on today, and I'll put them up in a separate file
Okay.
in the same heading under
Okay.
Moodle, so that you can see them. My advice would be to try and work them on your own before you go and look
I
at
just
the
wanted to
answers.
know, like, if I was doing it, if I was getting the right answers.
Yep, I told you last time I would put up an answer
Okay.
sheet. And I'll just put up an answer sheet for the first page, not the second page, since the second page deals with two-trait inheritance.
Okay.
We'll be covering that next week. What do you guys want to know? What are your questions? What do you not understand?
So I feel like we've done a really broad sense of a lot of different things.
Yeah.
And I was wondering if we could do like bullet pointed, like, these are the vague topics that are gonna be on the test, and then like
That's
kind
kind of
of
what we had
how deep.
on Wednesday.
Dude, I did that on Wednesday.
Okay, I wrote all that down, I didn't know if there was other stuff,
Um,
like going back to the omnivore's dilemma, what kind of.
Yeah, so in the omnivore's dilemma, for example, we talked about the national eating disorder. He talked about what the omnivore's dilemma is and
Yeah.
why it is a dilemma. So those kinds of things will be on there. I told you at the beginning of class that there will always be a question on the test. It's always the first question on every test. What is the omnivore's dilemma? You'd be amazed at how many people still miss that question on the final, which is why a number of years ago I just got in the habit of asking it on every test.
The omnivore's dilemma is for humans, there's so many different things you can eat that, how do you choose
What's the best
Yep.
option to
Yep.
choose?
When you can eat anything, how do you choose what you should eat versus what you shouldn't eat? Rats figure it out by looking at what kills or makes their fellow rats sick. Humans have not yet progressed to the intelligence level of rats. Rats will smell the breath and the fur of other rats when they come into the colony after they've been out foraging, and so they learn scents really well. And if a rat comes back and it smells a certain way and then it dies the next day, they'll associate that scent that came back on that rat with something that's bad, and then when they encounter that out in nature, they'll avoid that. If a rat comes back with a scent on its fur and that rat seems healthy, they'll file that away and say, okay, when I come into contact with that scent out in nature, I'll go ahead and eat that, whatever that was, because it can harm my individual. In animal behavior, it's called central place foraging, where all of the members of a colony go out and forage, and then they come all back to a central place. Well, when they come back to that central place, they're carrying useful information.
Do honeybees do that as well? Is that?
They don't do it that informally. They have a much more formal way of communicating the location of nectar sources. But honeybees are dietary specialists, so they don't have a dilemma. If it looks like honey, tastes like honey, they eat it.
Yeah.
In the same way that koala bears don't have a dilemma. If it looks like eucalyptus, if it tastes like eucalyptus, if it smells like eucalyptus, you eat.
Cool.
So just kind of like expanding on the stuff we read in the first chapter, so like corn walking.
Okay, so
We kind of went over
let's
that
go
with
through the book.
genetics, right?
So introduction.
Yeah.
Introduction is about basically two things: the national eating disorder, which is about a nation that is obsessed with healthy eating, that's one of the most unhealthy nations on the planet, and then what he defines what the omnivore's dilemma is and talks about where that comes from, looking at Rosen's work on rats, actually, the omnivore's dilemma, which I cannot seem to spell. So then in chapter 1, which we're still in chapter 1 of Pullen's book, he talks about corn walking. Well, what does that mean? That means that people, i.e. North Americans, have an isotopic signature that is made up of heavy carbon. And so if we took an isotopic sample from you, if we took a sample of tissues from you and ran stable isotopes on it, there's a lot more C13 and C14 in it. That heavy carbon isotopic signature is an isotopic signature that comes from C4 plants, which then got us into talking about different photosynthetic pathways. What are they? Well, there's the C3 pathway and the C4 pathway. And the CAM pathway, we didn't talk about CAM because CAM isn't found in widely used food sources. Those are things like cacti and things like that. But we did talk about the comparison and contrast of C3 and C4 because that's what leads to differences in isotopic signatures in organisms that eat plants or the organisms that eat other organisms that eat plants. And in order to understand this, we had to know something about plant anatomy, which includes plant anatomy of the cell, plant anatomy of the leaf, and plant anatomy of the plant body as a whole. The only reason we learned this is because C3 and C4 plants utilize different cells in doing C3 and C4 photosynthesis. And then we talked about carbon fixation, the beginning parts of photosynthesis, because this is the part that actually influences isotopic ratios. So all of that stuff about photosynthesis was in the service of you understanding that section of the first chapter where he talks about we are corn walking. Why are we corn walking? Because when we look at our isotopic signatures are the signatures of the C4 plant. And it doesn't have to just be corn, it can also be sugar, because sugarcane is also a C4 plant. But we have a lot of C4 plant signature in our isotopes because we're eating a lot of C4 plants, corn, cane sugar, things like that in our diet.
For the isotopic signature, like that heavy carbon, will we need to know like why that like the C12 carbons are processed much faster, or
They're processed faster because they're lighter.
okay.
And in a C3 plant, because they're constantly getting new carbon dioxide in, they process carbon 12 first, they leave carbon 13 and 14 behind, and they're always getting a new influx of carbon. C4 plants, they process all the carbon. They process the C12, they process C13, C14, it all gets converted into glucose, which is why they have a slightly heavier isotopic signature.
Okay.
And then the second part of this was dealing with corn sex. So there's a whole section of chapter 1 that deals with corn sex. Well, corn sex is all about, well, how did corn become the way it is? Well, corn became the way it is because it's actually selected by humans to become what it is. If you look at, we haven't gotten to this yet, but we'll get to it after we finish genetics. Corn came from a plant called teosinte. I haven't shown you this yet because we haven't gotten to genetic modification, because that's the next topic after genetics.
You can still find that in Mexico, right?
You can still find it in the wild in Mexico, as a matter of fact. Yep.
And I don't know how to pronounce it, the teosinte,
Mm-hmm.
that did not need man, right? Because there's like a chapter on married
Yeah.
man, so kind of produced by itself, reproduced on itself.
So like I said, we haven't gotten to this yet,
Yeah.
but this is teosinte, this is corn. So one of the questions that you should have from reading chapter 1 of Pullen is, how the hell did we get from this to that?
Yeah.
Well, we got that through actually a relatively small number of genetic modifications. They weren't genetic modifications like we think of genetic engineering in the 21st century. They were just simply selective breeding of corn plants. And so in order to understand selective breeding, which was responsible of taking teosinte to modern corn, we have to know how genetics works. In order to know how genetics works, we need to know about meiosis good way of studying meiosis is to think about mitosis, because the second meiotic division is just like mitosis. And also mitosis is just an important cellular process because it's how a baby corn seed gets to be a corn plant. In order to know about mitosis, meiosis, and to know about genetics, you need to know what a gene is. But genes by themselves aren't complete in knowing how you get a corn plant that looks like that, because genes get transcribed into RNA, which get translated into amino acid sequences, which are proteins, which leads to the phenotype. So in order to understand what a gene was, we had to know something about what DNA is and how it's structured. So once again, all of this stuff that we've been talking about in terms of mitosis, meiosis, DNA, central dogma, which is what this whole thing is, and genetics is all in the service of eventually, on second test material, understanding this transformation from a simple plant with shattering seeds that are released from the plant when they're ripe, to a plant that holds on to its seeds, is wrapped in these leaves, has lots of kernels as opposed to a relatively few number of kernels. How did we get from here to there? How do we get from a wolf to a pug? Well, that all involves all of this stuff here. And so we're going through this stuff not because I think that you need to know about genetics. It's just that in order to understand the kinds of things that Poland is talking about, you need to understand genetics. And so this is what was on the board basically from Wednesday at the beginning of class. This is, because somebody asked, oh, can you provide us with a study sheet? And so I just provided you with a study sheet. So that's what's going to be on the test. And I have 100 points to get that information out of you. Yeah.
Regarding like the actual biology of the plants, like the plant body, how in depth are we going to need to know like about the leaf structure?
I'm going to do exactly what I said last time, which is I'm going to put a figure of the leaf structure that comes from your PowerPoints on there, and I'm going to just blank out the names of things. Once again, concentrate on leaf structure in terms of those things that are actually important for the function of the plant. So knowing about the mesophyll cells versus the bundle sheath cells. Well, the bundle sheath cells are around the vascular bundles. Oh, well, we talked about vascular bundles because vascular bundles are important in moving materials around the plant, xylem and phloem, moving either materials from the roots up to the leaves, or moving materials, the results of photosynthesis actually, from the leaves to be stored down in the roots. So the vascular bundles, xylem and phloem, their arrangement in the plant leaf, mesophyll cells, bundle sheath cells, stomata, where are the stomata located, what do they look like, how do they function? Those are all things that have to do with photosynthesis and fluid movement around the plant. Other stuff about the plant body, I'm not really going to worry about that much because it's not really relevant to the discussion of photosynthesis. Now we talked about floral structure, because floral structure is once again important in reproduction. So learning those parts of the flower, stamens and anthers and pistils and the ovules and all that stuff. Knowing the difference between imperfect and perfect flowers. Perfect flowers having male and female parts, imperfect flowers having male and female parts in different flowers. That prevents inbreeding. What were we talking about? When you inbreed over and over again, you tend to get homozygosity dominating in plant populations, and homozygosity leads to lower genetic diversity. And so imperfect flowers are actually good to prevent inbreeding, which is probably why some species have imperfect flowers. You didn't know why I was giving you imperfect flowers back at the beginning, but now that we've talked about inbreeding, you can go back and connect. Oh, we talked about inbreeding, how that leads to a bunch of homozygous traits in plants, and imperfect flowers are one of the ways that you get rid of that. So don't forget to connect information across what you've learned. Because imperfect flowers seem weird. Why would you put the male parts in one flower and the female parts in another flower? Why not put them together? Then you're guaranteed fertilization, But the problem with guaranteed fertilization is it also leads to inbreeding. What else? Does that help?
Yeah.
Thomas?
Mm-hmm.
So parse the material. Ask yourself, of this figure that is in this PowerPoint, what of that figure was important for you? They're understanding genetics from reproduction, and what in that figure is necessary to understand photosynthesis.
So would you say the best way to study is going through the PowerPoints, figuring out what is important for this slide,
Sure.
memorize, move on?
Yeah, I don't make a PowerPoint unless it's useful in me getting across the information to you. Yeah, and pay attention to the things that I have paid attention to in the PowerPoint. So let's go back to the basic plant biology part.
So is corn it's a grass flower, so it's incomplete but perfect, correct? 'Cause it's got a pistil and a stamen, they're just not.
So the flowers are incomplete because they lack things like petals.
Mm-hmm.
But they're also imperfect because there are male flowers and female flowers. Remember, the male flowers are the tassel at the top.
Yeah.
Those are the stamens. And then the ear of the corn is about midway down the stalk. Those are the female flowers. When you eat corn on the cob and you shuck your corn and there are all those threads, the silk of the plant, those silk are actually the pistils
Okay.
of the plant. And so the male flowers are located at the top of the plant, the female flowers are located about halfway down the plant. So those flowers are not only incomplete, but they're also imperfect because you have male flowers and female flowers and they're separate.
Okay.
And I did say that in class, but I might have blown through
So
it.
is
pretty
corn
fast.
like a one house? Is that monoecious then?
So it's one house because both male and female flowers are on the same plant, as opposed to a ginkgo tree where a whole tree will have nothing but female flowers and another whole tree will have nothing but male flowers. And the flowers that you see, the trees that you see that are in people's gardens and in yards and stuff, those are all, I think they're female trees because the male flower stinks to high heaven. To attract pollinators, presumably, but they smell really bad, and so most of the trees, the ginkgo trees that you see, I think it's the female flower that doesn't smell bad. Those are dioecious because you have the male and female flowers actually in two houses on separate plants. Let's go to.
Have you ever heard of the rainbow tree? The one that has like the bark that's all different types of colors?
Oh yeah, it's a type of eucalyptus. It's really common in Hawaii. It's pretty.
Those, do you have any clue if those would grow well in the like Midwest?
No, they're a tropics, subtropical species, and so it would get too cold, it would probably kill them in the winter here. So, we talked about vascular bundles and how the shapes are different. That's mainly so that you can separate the difference between monocots, things like grasses, which is what corn is, and dicots, things like soybeans, because those are two important plant species that we talk about. So knowing vascular bundle structure is important, but what's probably most important is that you'd be able to identify two things in it, xylem and phloem, because those are the vascular tissues that are responsible for the movement of fluids up and down the plant, and that you'd be able to recognize what a vascular bundle is in a monocot and what a vascular bundle is in a dicot. They look different when you look at them individually, and they look different when you look at them in terms of their distribution. In monocots, they're spread throughout the ground tissue, and in dicots, they're located in a ring. But once again, that's just so that you can recognize the difference between monocots and dicots, these two big divisions of plant structure, general plant structure. And once again, when we talk about the structure of the leaf, there are only certain parts of it that you really need to think about. We talked about the cuticle, because that's a waxy coating that helps hold in water. We talked about xylem and phloem, we talked about bundle tree cells, we talked about mesophyll cells, and we talked about guard cells. We didn't talk about the difference between palisade and spongy mesophyll because it doesn't really matter, because those are all mesophyll cells. From the standpoint of thinking about them from the perspective of photosynthesis, there's no difference between a palisade mesophyll and spongy mesophyll. They all do photosynthesis. So So I didn't talk about these divisions because it's not important from the standpoint of what I'm concentrating on. Does that make sense?
What does the guard cell do again? Does that allow?
The guard cells open and close. Here's a closed
that's
guard
a
cell.
stomata.
Here's a more open guard cell. They're the things that open and close to let carbon dioxide in. When they're open, they let carbon dioxide in, but they also let water out. And then remember that there's this animation that is, once again, more useful to, well, what happened?
So they just regulate the opening and closing of the stomata.
What happened to the rest of this? Oh, I know, it's in the photosynthesis. Yeah, so the guard cells regulate the opening and closing of the stomata. The stomata, a stoma, is the hole that is made by guard cells.
Okay.
Yeah. And so in the,
We're gonna go to the animation where
Yeah,
it just
the
opens up
photosynthesis.
and it flows in.
Yep.
Okay.
So when you then think about that, think about it in the context of photosynthesis. So the only reason you need to know about guard cells is because guard cells are important in this. And so this is one of those situations where you would want to actually have the PowerPoint and not just images, because dimmer things represent lower concentration, bold things represent higher concentrations. During photosynthesis, carbon dioxide is picked up by rubisco, incorporated into glucose. As that happens, concentration of CO2 goes down until it finally gets low enough that oxygen starts interacting with rubisco. But before that happens in a C3 plant, you don't want that to happen because that's a less efficient way of going about getting glucose. You want to actually, instead of having that happen, you want your stomata to open. So those are the guard cells. I have drawn them in as closed and then got rid of my little circles to show them being open. Once you open that up, carbon dioxide can flow in, restoring CO2 levels inside the leaf, but water leaves because water is at a higher concentration inside the cell than outside the cell because it's around all these nice cells. Then the stomata close themselves up and then they start fixing all that carbon dioxide that they just brought in. But for a plant to conserve water, it needs to open its stomata less often and for shorter periods of time. And if you don't have a way of keeping rubisco away from oxygen, you have to constantly be opening your cells. C4 plants can conserve water because they've hit upon a way of keeping rubisco protected from oxygen by not having rubisco in all the mesophyll cells. They just have the rubisco in the bundle G cells. In the mesophyll cells, carbon is initially captured by PEP carboxylase, which only interacts with carbon dioxide, as opposed to rubisco, which interacts with carbon dioxide and oxygen.
Could you see that being a short answer on the test?
Oh, yeah. I mean, how many days did we spend on this?
Multiple.
Yeah, yeah. So people are always like, oh, well, what's going to be on the test? I'm like, well, what did we spend time on?
Mm-hmm.
That's what's going to be on the test. Sure. Yeah.
What's the little circle for on the right side? What is that?
Oh, that's just
What
it's
is
a
the circle?
it's a figure from the book.
Okay, I just didn't know like what those were.
This circle goes to a line.
Okay.
This is that same image.
Oh, okay.
It's just blowing up C4 photosynthesis to show that initial carbon fixation goes on in the mesophyll cells, and then you use PEP carboxylase capturing CO2, adding it to phospholinopyruvate to create malic acid. Malic acid then gets transferred into the bundle sheath cells where you strip off a CO2 and then just run it into rubisco, into the Calvin cycle to make glucose. But the rubisco, whereas there's rubisco there, that's held in the bundle sheath cells that have these thick coatings around it that keep both oxygen and carbon dioxide out. The carbon dioxide gets in because it gets shunted in on this carrier that is malic acid.
Okay.
So this was the thing where we talked about the mail guy
Mm-hmm.
bringing mail up to all of the offices. The mail comes in the basement of a newspaper, then gets sorted into different baskets, then it gets delivered to the office it needs to go to. Malic acid is kind of like the basket that carries CO2 into the bundle sheath cell. So that
Okay.
this figure is just a blow up
Mhm.
of this figure.
Gotcha.
Yeah.
Okay.
I wouldn't have either of those figures in here if I didn't think they helped you understand, in this case, the spatial separation of initial carbon fixation versus the generation of glucose in mesophyll cells and bundle sheath cells, which is different in C4 plants. That's the way it is in C4 plants, as opposed to C3 plants where all of this just happens in the mesophyll cells. But the mesophyll cells are out there and they're exposed to oxygen. This mesophyll cell is exposed to oxygen, but it doesn't matter because if you deplete CO2 levels out here, pentacarboxylase doesn't interact with oxygen. What else? Ooh.
That's about it for me.
Study. I don't know that, I didn't like write stuff down.
Okay.
That's not anything that.
Yeah,
Retrieval.
I find recopying things and trying to say, okay, if he asked me a short answer question on a test, can I draw a mesophyll cell sitting next to a bundle sheath cell and kind of show where the things are? Where's PEP carboxylase? Where's rubisco? Where's carbon dioxide? What does PEP carboxylase bind to? You don't have to draw it in a fancy pants way like this. You can also draw it in simpler schematics like this. This is a simple comparison between C3 photosynthesis and C4 photosynthesis. The whole purpose of this is to take something more complicated, break it down to something that's more schematic, if you think that way about it. Okay?
Cool.
A dozen ways to skin a cat. Figure out the way that works for you. And if you're having trouble with it, come by and see me between now and the end of the day today.
Okay. Sounds good. Thank you.
Email me over the weekend if you want. I'm online somewhat over the weekend, but if you have a burning question that you don't know the answer to, just shoot me an email.
All right. Sounds good. Have a good weekend.
I'll
Thank
often
you.
reply with an answer to everybody in the class so that everybody has the answer, so that if somebody else had the question and just didn't want to ask, they have access to it. So they'll know that you had the question, but it's probably beneficial for them to see the answer. Yeah.
Great. All right. Have a good weekend.
Yep, you too.